All right, we're going to start at the top of the hour here. Welcome to State of the Science. State of the Science Bioimaging. And we are very happy to have a very esteemed panel here. We have Dr. Mark O. Huising, PhD, professor at UC Davis. He will chair this panel. Vira Kravets, PhD, assistant professor, Department of Bioengineering, Department of Pediatrics at UCSD in California. Julia Panzer, PhD, she's a senior scientist at City of Hope in Los Angeles, California. Adrian Villalba Felipe, PhD, is a postdoctoral fellow at INSERM Paris, France. Jack Virostko, PhD, assistant professor at Diagnostic Medicine, Investigation and Inquiry at Dell Medical School, UTexas, Austin in Texas. And Stan Jankauskas, PhD, a postdoctoral fellow at Albert Einstein College of Medicine in New York City. Finally, we also have joining us Nicholas Mamrak, PhD, is a scientist in cell therapy at JDRF, New York City in New York. And this will be a sort of a 1-hour panel to talk about the challenges, progress, and roadmap to full clarification of human pancreatic function in context of type 1 diabetes. You can see a quick access to all panelists linked in the chat. And if you need further information on their bios, that's where it is. So welcome and thank you all for joining us.
All right, so I'll second that. Thank you, Monica, for putting us together. This is awesome. It is really nice to have people together who really are trying to push the envelope as where we are when it comes to imaging the pancreas at different scales, at different time resolutions. And I think this is a wonderful opportunity for us to get together and just learn from our colleagues where we are, what we are capable of doing when it comes to imaging the pancreas. Personally, I think I'm not alone in sharing that I'm a visual learner. I think there's nothing like seeing the data, seeing what's happening, and seeing the processes that we study, sometimes in real time, sometimes over time, longitudinally is I think a very powerful way of track the pancreas, both in a healthy as well as in the type 1 diabetes state. So before we are going to get treated on a couple of vignettes of what the panelists each are doing to further their own questions, we're going to have a short discussion around where we are in imaging and where we might be going. And there's a couple of questions that I'll put in the chat while I post them, or maybe Monica or somebody else can put them in the chat. The first question I want to pose to the panel is, what is the imaging challenge that you're working to resolve right now? As soon as we get off the call, what are you going to go back and try and get to work that is just that close, but not quite there? Julia, please.
Yeah. So, I mean, at the moment, we are trying to figure out how we can label cells specifically. So that I think is still a big challenge. So we have all kinds of dyes to label them because they're viable cells, right? So you don't have that many options. And I think we're just not there yet. So there are so many different cells that we're trying to look at, and to specifically label one population, that would be amazing. So that's what we are currently working on.
Thanks for sharing. And yeah, that is a big one. And it's easy to do it in mouse, relatively speaking. And it's much, much harder to do it in humans. In our experience, in human islets, there is just a lot more heterogeneity extending to the point that we can always be sure that certain responses that are textbook alpha cell or textbook beta cell are indeed uniquely reserved for the cell type that we typically associate them with. And I'm sure that's the experience of Julia as well, because that's you're nodding. Anybody else on the panel want to chime in what they're currently working on?
I can jump in there as kind of a non-microscopy person. So I'm a MR scientist. And so when I think of imaging the pancreas, I think of imaging, the whole pancreas in an intact human, but our images look nothing like the beautiful images you're going to see here, right? So we kind of see, the gross structure of the pancreas. We can see some microstructure, but I really want to kind of push the resolution and kind of the contrast of what we're doing in the MR world to be able to, we're certainly never going to get to kind of the resolution and the sensitivity of optics, but at least to be able to kind of start to bridge those gaps. I see this kind of big chasm between, a lot of what you're going to see in microscopy and what we can do in vivo. And I really want to kind of see how we can kind of push those worlds closer together.
Yeah, thanks for sharing that. Plus, the imaging that you do is actually close to home, because that's actually imaging the pancreas of patients and longitudinally. So in many ways, the most meaningful way to try and visualize the pancreas. And a final person from the panel want to chime in? What are they working on?
Yeah, I would like to share that I think it would be nice to have more availability to label multiple markers in the same sample. Now we have this kind of maxima devices, and those that are using nanobodies labeled conjugated to a very specific target that can be bleached extremely in few minutes. But this device is not yet available for many countries and for many institutes, and I think it's one of the next barriers that we are going to cross in the early future.
Thanks, Adrian, for sharing that. And that's actually a very good segue to the next question. What type of imaging do you hope you, or maybe the field more widely, is capable of doing 5 years from now? Like think about what you're doing now, and wishful thinking, like where do we hope to be in just a couple of years, to really, when we might look back at what we're doing now and say, oh, remember that 5 years ago we were doing this, and now look at how far we've come. Anybody want to tackle that question and share their thoughts on that?
Maybe I'll start. So in our lab, we try to do multimodal imaging, and we image living tissue slices from pancreatic donors. And right now we're practicing with mice to image calcium dynamics and also phlegm, use an ADH lifetime to see the metabolic activity and how does that change. So 5 years from now, I hope to be able to use that to do a label-free cell identification based on metabolic activity of the cells, and I have reason to believe that this is possible. And then on top of that, the dream is to also image the immune cell interaction, the contact-based interactions in the type 1 sample, which is possible to do in vivo in mice, but it would be amazing to also do this in pancreatic slices. And if we could do it in our lab, that would be a dream come true.
Excellent. Thank you, Vira. I look forward to seeing that, because I know that's a very challenging goal. Anybody else on the panel want to share where?
Yeah, I can chime in. So I think from a funding perspective, the way we look at things, sort of broader buckets, sort of landscape view, I think what we're really excited about is translation, right? So whether that's the really great MRI work that Jack's working on, or you know, some of the PET or SPEC probes kind of moving to prime time in a clinical setting, you know, I think that's what gets us really excited, because we can apply that then, you know, whether it's natural disease progression, looking at how beta cell mass changes throughout the course of disease, or maybe in a cell transplant setting. You know, these come from both extremely interesting biological questions, but then having diagnostic and, you know, diagnostic, we start thinking about actually applying these tools. So I think that's, you know, hopefully 5 years we can get us there.
Yeah, thanks. And thanks for sharing, like, ultimately, like, the big picture funding perspective, because the challenges that hopefully we come up with today will require funding and effort to solve. So these discussions, I think, are super helpful to try and coalesce around, like, some of the challenges that are ahead of us, so we can direct effort, both financial and people and brain-wise, to try and solve these challenges. Thanks for sharing that, Nicholas. Anybody else? 5 years from now, where do we hope to be?
Maybe not in 5 years, because it's a short period of time, but I would like to see someday vivo bioimaging of the human pancreas in patients from a clinical viewpoint. But I think it's quite utopic and unrealistic to think this will happen in 5 years, but maybe much more this would be feasible. Who knows?
Yeah, I think that's kind of the holy grail. If you can do that, I mean, we would be able to just directly just focus on just patients and skip over all the stepping stones we have taken to get there. Maybe I'll share from my perspective what we are increasingly capable of doing is leveraging the ability to image dynamic signaling, multiple signaling cascades and multiple cell types in real time, see how they interact, and then quantify the signaling cascades, because we talk a lot about heterogeneity amongst alpha cells and beta cells, and that's certainly very true, but a lot of that work is grounded in transcriptomes, And I think approaching that from the actual functional responses up, where I think the ultimate functional response would be direct secretion of hormones. I think we're a ways away from doing that at scale. But I think that will be a challenge that is super exciting to try and meet. Okay, so then let's talk about what's holding us back. What is the biggest barrier? What is a question that right now you can't answer, even though you wish you were able to? What do we need to direct efforts and resources to try and fix?
Okay, so I work a lot with mitochondria, and I visualize mitochondria in the beta cells. And the main challenge that I face is that the majority of the standard programs for visualizing, they are very bad in capturing mitochondrial morphology. They are very bad in distinguishing between different mitochondria. And I would really like if there will be some maybe IE-based tool that will be, much better in recognizing different organelles. So that's that's the main challenge I face, in my project.
So let me expand on that a little bit. Is the challenge here the quality of the image that you're acquiring, or is the challenge the quality of the analysis pipeline that you have to use to then do the post-hoc, post-imaging analysis?
Is the challenge for the quantification. So of course, so when I took image and by eye, I can clearly see that this is one mitochondria, this is another mitochondria. It's very difficult to find an algorithm in the majority of the programs that we use for quantification to finally see the structure of mitochondria, because we all know that beta cells have high heterogeneity, but inside beta cells, mitochondria have also heterogeneity. It's very interesting to measure the median parameters of the whole mitochondria in the cells, but to see how different subpopulations of mitochondria are changing in the beta cells.
Great, thanks for clarifying that. I understand this.
And Mark, I'd echo something you made in your prior comments, and that's I think you mentioned the word quantification, right? So how do we quantify the signal, right? And not only how do we quantify it at one single time point, but how do we make it repeatable and reproducible so that we can make that same measurement on our own experimental system, months, years down the line? How can we make that same sort of measurement as a different lab, perhaps using different instrumentation? I'm kind of acutely aware of how difficult that can be in the MR world. I'm certain there's a lot of analogous problems in the microscopy optical imaging world as well.
Yeah, excellent comment. Fully agree. Thank you, Jack. Anybody else? A barrier?
Quick question in the chat from Alejandro Sacaido. Any plans for data sharing platforms akin to the mRNA databases?
Yeah, I think that's an excellent point. There are actually some image data sharing platforms available, and journals increasingly are requesting that one deposits, and I think that's appropriate. And the extension of that is that, and I've seen this over the last decade or so when sequencing data became available, is we should start using those data, right? We still, on the sequencing side and the imaging side, it's probably the same thing. It's still not as common as it should be that instead of doing the experiment ourselves, we're basically just taking the experiment that somebody else did maybe a couple of years ago, but it may be no better than what you would do yourself. So rather than repeating somebody else's experiment, just use somebody's data as your first go-to. And the same thing should extend to imaging. If I have interest in imaging calcium dynamics in human slices, I should be able to pull Julia's or Vira's data as a starting point, rather than just try and do it myself without reanalyzing some of these data. So yeah, Alejandro, I fully agree. I think we should, as a courtesy and as a habit, continue the practice of sharing our imaging data. And some of these platforms are becoming available. And yes, this is also a barrier. So I think as we are generating larger volumes of data, both in the storage and maintenance side in our labs, as well as in how do we share this freely, there is challenges that need to be continuously met as the data sets grow larger and larger. Okay, anybody else want to chime in on Alejo's comment? Because I think it's a really important one.
I would just ask, what are your thoughts on the pancreatless? Because that whole effort is really aimed at that. One of their endpoints is to create something like that, correct?
That is correct. And I think that's fantastic. I think NIH mandated that. And I think the HERN consortium and the folks at Vanderbilt really, I think, stepped in and leaned in and started depositing lots of data there. And I think we should get in the habit of also using those data, because I think that's where I think as a field we can grow.
Yeah, I was going to say we actually used some of their data already, and my students are working on quantifying contact-based interactions with alpha-linked beta cells. So I'll show something that we found in mice and that we just published it, and then I'll tell you what we found using PANCreaTless. So it's amazing that there's immunofluorescence data already there, but what is lacking is a dynamic data, which is, I think, a lot of us are doing, the calcium dynamics. That would be really good. A quality, high resolution, temporal resolution data is definitely lacking.
Wonderful. Thanks for sharing, Vera. That's awesome to hear. All right, we have a few minutes left before we're going to start with videos. So final question for now. And this is informed by how I have run the science in my lab. I have found it really helpful to leverage tools people have developed for me. I'm not a tool developer. I wish I was. I'm just not good enough. But I have graciously used tools that others have developed in our field or in other fields for me. So maybe that informs the question. Like, if you could ask any tool that would help you, what is the tool that you wish somebody would develop for you so you could apply it to the pancreas?
My favorite tool is really easily accessible human tissue slices.
Yes, I can second that. Actually, NPOD has been super helpful in sharing slices. So I think what our field has done really well in general, and I think NPOD has been leading the way along with the IADP, is come together and organize these biorepositories. I think other fields have not done this nearly as well as we have. So the folks on the call who have led that, I mean, I know Alberto was here. I mean, deeply grateful for the field just having organized itself to make that a reality, because I think it's really made a large difference in the last decade and a half. So just a shout out for that. All right, one more tool. Who wants to go in? What's the tool you would want? Maybe somebody's listening and actually will develop it for you. So here's your chance.
I would like to have either a universal format for each image or an easy converter from one software to another, because sometimes images are generated in raw data from one microscope that cannot be analyzed by one software if you do not follow 3 steps of conversion that take a lot of time. So I think that an easy to use converter or either a universal format ideally would be nice.
Yeah, data uniformity and data formats certainly would make our jobs a lot easier. Okay, so with that, we're going to switch over into the panelists, each sharing a very brief video to kind of give the group a flavor of the type of imaging that's going on right now. And at the end of that session, we're going to have a discussion that can go anywhere the group wants to go. I'll start. I'll share my screen, and we'll basically like give a 2-minute high overview primer of the type of stuff that we do. It's really interested in understanding the complex crosstalk between the main islet site cell types, alpha and beta and delta cells within the islet. But if you were to extend it to slices, how exocrine and ductal cells contribute to this, and how dynamically this changes during an immune challenge. We all know that beta cells are releasing insulin, but they're doing so under control of various paracrine neural and nutrient stimuli to regulate or sometimes dysregulate the insulin and the glucagon that's coming out of these islets, depending on whether we're looking at a healthy or a diseased pancreas. And this is governed by a large array of receptors. This is just the GPCRs that are all there, all doing something. For some of them, we understand quite well what they're doing and through what cell type they are engaging. But for other targets, we probably don't know much, if anything at all, about what the role is of these receptors that are expressed. We just know that more than likely there is a role that we just haven't understood yet. And it indicates the complexity of just a 3 endocrine cell system that normally in a healthy pancreas controls beta cell insulin secretion really appropriately and carefully across about 80,000 meals that we hope to ingest over a lifetime. Controlling just a simple system like that takes hundreds of receptors that are each coupled to different GPCRs. And we really only, I think, understand like a small fraction of the complex biology that is going on in what in essence is just still a fairly simple system. And what we're trying to do in our lab and getting better and better at is to image multiple different signaling cascades. We're mostly focusing on calcium and cyclic AMP because we can, and because they're important signaling pathways, obviously. And we purposefully are looking at multiple cell types at the same time in real time, because what it affords us is the ability to actually observe and quantify their dynamic behaviors, both when it comes to different cell types and as it relates to 2 different signaling cascades in the same cell at the same time. The image I showed you here was mice, because everything is easier in mice. We can do this in humans as well. And we can also really start to leverage scale if we take that and instead of doing it in intact islets, do it in a dissociated format, where we are able to, in a single imaging trace, quantify the signaling responses of, in this case, both calcium and cyclic AMP in real time across a couple of thousand of beta and alpha cells, and then resolve after the imaging what cell type was which by doing a simple post-hoc fluorescence. And the bottleneck here then becomes, how do you automatically segment these cells and get the data out? Because the scales at which we're doing this is no longer feasible to draw manual ROI. So we find that we spend a lot of time trying to solve that bottleneck rather than the actual imaging itself. So that was just a primer of what we're doing. Next, Vira, do you want to go next? I apologize, there's actually a plane flying over. I hope you can't hear it on your end, but if you do, my apologies.
Okay. I hope everybody can hear me and see my slides. Yeah.
Yes,
Okay.
perfectly.
So let's go. So glucose homeostasis is supported by multiple cell types, but also interaction between the cell types, as was just mentioned. And the 2 things that we study in my lab is electrical and paracrine interaction. So paracrine, mostly we focus right now on alpha beta cell communication. There's also neural interaction, which I would love to dive into, and this is a picture from Alejandro Caicedo's lab, but we haven't gotten there yet. And so in human tissue slices, which are provided by NPOD, I was lucky enough to collect some data when I was in Richard Benninger's lab, where I would look at the calcium dynamics, which is shown here in orange, in pancreatic islets from human tissue donors. And then from that, we could post-stain these islets and identify which cells are insulin-secreting beta cells and which cells are alpha and delta cells. And then the dynamics of these cells are shown here, where non-beta cells usually, but not always, are more active at low glucose and less active at high, versus beta cells. They have this characteristic low activity at low glucose, and then first phase response and second phase response to glucose elevation. So this first phase of response is really interesting because it is lost in pre-type 1 diabetes at the point where type 1 has not developed just yet. And then it's completely gone in type 1 diabetes patients. And why, it's unknown. So the hypothesis that I have in my research is that it's because of the loss of the first responder beta cells. These are the beta cells that respond to glucose sooner than the rest. And let's see if the video is going to play. If you look at the calcium traces of these cells, these would be the first responder beta cells. And we also find that alpha beta cell communication is important because when you do the imaging in a live islet and then you post-stain the same islet for hormones, we find that first responder beta cells tend to be near alpha cells. And we can then overlay the 2 images and identify the same islet plane and tell that which cells that respond sooner to glucose located near which other cell types. So we find that alpha cells tend to be near first responder beta cells much more than near the last responder beta cell. So this has got us thinking is that our beta cells in the alpha cell reach areas of islets, which are likely first responders, targeted first in type 1 diabetes. Could it be that these first responder cells are killed first? And this is why we have a loss of first phase insulin secretion. So we attacked this question by looking at first NOD mouse model, and it just got published in 2024. The abstract is online. We found that based on the contact-based structural network, the alpha cell regions or regions where beta cells are alpha cell linked, as opposed to beta cells that don't have alpha cell neighbors, there is much more T cell interactions compared to if we take this network and we randomize the positions of the immune cells. And so this would be the shuffled network. There's much more interactions in alpha cell region. So it says that alpha cells may be playing a role of either enticing the beta cells to work harder and attract immune cells, or perhaps alpha cells dysfunction is attracting immune cells. So we don't know what's going on, but we would love to figure it out. And we are now looking at the pancreatic data where we've seen, at least preliminary, that there's a similar pattern, even though it's much harder to detangle because there's alpha and beta cell interactions much more prevalent than in this. Thank you.
That's amazing, Vera. Thank you for sharing that. We'll hold questions until after we are done with the video. So next person is Julia Panzer, City of Hope.
Okay, so I can kind of piggyback a little bit on Vira and Mark because we're also doing calcium imaging a lot. And like Vira, we're also using pancreatic tissue slices. And you can see like a quarter of a tissue slice shown here. So before we start imaging, we usually perform a little tile scan to just see where we're at in the tissue. And these two here are the harp strings, which is a harp that we use to kind of press down the tissue so it's not moving during imaging. And then you can see that there are actually many islets inside the tissue, and that's the great part of the slice. So you can actually look at different islets. You can look at them in different positions, close to other cell types, like for example, close to neurons, the blood vessel, and also you can look at the immune cells, which are still there. And also you can look at small clusters or bigger cells. So then we basically look at individual islets, and then we can either look at one individual one in different planes and in more detail and higher resolution, or we can also image multiple islets in the same slice. And then very similar to what Mark and Vera showed, then we perform calcium imaging. And as I said in the very beginning, we use unspecific dyes because this is human tissue. So we're still working on labeling very specific cell types reliably so that we can look at the entire cell population. And then after performing this imaging, we can create all kinds of analysis. So most common by now is to create these heat maps. And because of all the stimuli that we apply to the tissue, we can then sort the cells. So for example, I'm very interested in looking at alpha cells. And that's another advantage of the slice platform because alpha cells are still there in type 1 diabetes. But if you isolate the cells, then many of these destroyed clustered might not survive this process. But in the slice, we can actually look at them. And then we can see what happens in type 1 diabetes and how their function is lost. And another thing that we're trying to do is to create these response maps. So by having these different stimuli, and these are now all alpha cells, we can basically sort out the ones that respond to glucose or to kainate or adrenaline as a stimulus, and then create these response maps, which kind of give us a functional atlas of the islet. And coming back to the discussion that we have, I think if there would be a tool now to pick these individual cells and tell me, you know, the transcriptomic profiles of these in 3D, that would just be amazing.
Thank you, Julia. And yes, that's maybe a barrier that we can spend some more time talking about because I think that's a very real one that might take some effort to overcome. Next, Adrian, who's joining us from Paris. Thank you for doing that. I'm sure it's late on your end, so I appreciate you being here.
Thanks to you. So I want to begin saying that I really like the title that Monica decided for this session, because we have already discussed about challenges, promises, and now we can talk a little bit about the part of clarification, because I will show you our approach, which was recently published in Diabetologia this spring, in which we combine in toto staining of human fetal pancreases from PC week, post-conceptional week 5 till 12, the first trimester of pregnancy, clearing, which is the process in which we are able to remove, by delipidation, to remove the lipidic part of the pancreas, and then to label with antibodies in toto, the full specimen. And we are able to image samples like this one, which have the size of, this is a PC week 8 embryo, human embryo with in magenta the highlighted pancreas here. And at this moment, we are only able to image this kind of size of pancreas, but hopefully one day, maybe in 5 years, we are able to do so in full intact human adult pancreases. So here I showed you that I will show you 3 advantages of this technique. The first one, here we have the SOX9 positive epithelium, which is not restricted only to the duct in the human fetal pancreas. It stains the full epithelial tree. And in white, we have the insulin, a lignet to the center. And here I want to link it with the amazing paper from Ulf Algren, who is unfortunately unable to join us today, to show that this approach might be useful to image the full content of insulin. And this would have a lot of relations and a lot of reports with type 1 diabetes, in which we are interested in knowing which kind of islets are surviving to the autoimmune attack, or which kind of islets are still responsive to insulin secretion. The second main advantage that could have this technique is that we are able to discover new extrapancreatic connections. Here we show that the arteries are not yet irrigating the pancreas, but that blood vessels with this specific marker, CD34, are homogeneously distributed across the full epithelium. I think that for type 1 diabetes, if we would be able to image even a proportion or a small portion of the pancreas, this could be extremely interesting, not only with vessels, neurons, and these kind of systems, but also with immune cells and other of peripheral tissues that interact and are relevant for diabetes initiation. And lastly, with this technique, we were able to identify, you will see here, some insulin positive cells that are also in the duct draining the gut, and also in the gut part, which is not new at all. We have another paper from the team of Itzkovich showing this in human, but I think this approach would be interesting to identify other rare but significant cell types. We know about some alpha cells that might transdifferentiate into beta cell upon specific treatments. We know that there are virgin beta cells in the periphery of the islets that have or might have an important role both in physiology and pathology. And we also have this never-ending question about the existence of a specific pancreatic or endocrine progenitors in the human or even in the adult human pancreas, that if we would be able to identify by this in toto approach all the cell types using specific markers, could be very interesting. Thank you.
Amazing. Thank you so much for sharing that. Next is Jack Virostko at Dell Medical School at UTexas Austin.
All right, I pre-recorded audio to keep myself on time here, but if there's any issues with the audio, please someone speak up and let me know, and I'll go through the slides. I'm Jack Virostko, and I'm excited to talk about what MRI can tell us about the pancreas of individuals with type 1 diabetes. We've known for some time that the pancreas is smaller in people with type 1 diabetes, but many may not appreciate just how much smaller. The pancreas of the 10-year-old boy on the left is about twice as large as the boy on the right, who was recently diagnosed with type 1 diabetes. Pancreas size is also smaller prior to diagnosis with T1D. Individuals with autoantibodies conveying risk for T1D have a smaller pancreas. Pancreas size declines as subjects progress to higher stages of T1D. And longitudinal scans of the same individuals display declines in pancreas size beginning 3 to 4 years prior to diagnosis with stage 3 or treated T1D. Interestingly, pancreas size does not tend to correlate with oral glucose tolerance testing, where the two measures combined may be more predictive than either measurement alone. To better understand why pancreas size is smaller, we performed MRI of a family with permanent neonatal diabetes. The father and 3 of his children carry a mutation in their insulin gene where they no longer make insulin, but without the autoimmunity of T1D. All individuals who did not make insulin had a small pancreas, suggesting that insulin deficiency can cause small pancreas size. MRI can tell us much beyond the size of the pancreas. MRI can show us the shape of the pancreas, which tends to be thinner in people with T1D. Techniques like diffusion-weighted MRI reveal areas in the pancreas of people with T1D that may have increased inflammation. The MRI we acquire of the pancreas is currently much poorer quality than that is what is achievable in the brain, such as this beautiful brain MRI acquired in April of this year, which shows exquisite details of brain regions and blood vessels. However, if one looks at early brain MRI, such as this scan from the 1980s, one can appreciate the technological improvements that may be possible. MRI of the pancreas may never reach the quality of the brain, which benefits from a rigid skull. In contrast, pancreas MRI suffers from respiratory motion and blood flow artifacts. However, as seen in this ex vivo MRI of the human fetal pancreas, substructures such as the main pancreatic duct and its walls can be visualized. One of the lessons we've learned from fields such as neurology and oncology, where MRI has clinical impact, is the need to standardize measurements. MRI scanner settings and hardware can influence MRI measurements and must be carefully controlled. Toward this end, we've created a standardized MRI protocol for quantitative pancreas MRI and implemented it at 5 sites. We've assessed the reproducibility of pancreas MRI using MRI phantoms shipped to each site and from 5 controls who travel to the US sites. The protocol is shared on GitHub. One of the barriers to widespread implementation of pancreas MRI is cost and availability. However, low-cost portable MRI scanners have been developed and cited in sub-Saharan Africa. Artificial intelligence can read MRI images and quantify pancreas volume similar to a human reader, obviating the need for radiologist interpretation. In the future, MRI may become a part of well-child visits to image brain development and screen for suspicious lesions. I hope that pancreas screening for diabetic risk can be a part of such screening. Thank you very much for your attention. Please feel free to email with any questions.
Jack, that was amazing. It was right on time. Thank you for that. And I always think that neuroscientists have had an advantage because they have the benefit of a skull that we, I think, for imaging purposes alone, are sorely lacking. So, yeah, that was really amazing, and to see how far the field has come in being able to actually quantify pancreas volume and pancreas size in people with diabetes. Last but not least, of the panelists, we'll share a snapshot of what they're working on. Stan Jankauskas, who's at Einstein University in New York.
Okay. So I do a lot of live imaging to study the function of mitochondria, and today I will shortly give the example how I use live imaging to study mitophagy type 1 diabetes. So I ended up with studying mitophagy in my project in which I was able to delay onset of type 1 diabetes in NOD mice with a ketone body BHB. And from RNA-seq, I found that the protective effect most likely is mediated by the upregulation of optineurin, which plays a crucial role in mitophagy. And I confirmed that BHB really upregulates optineurin in pancreases of NOD mice, and there is more colocalization of mitochondria with optineurin. And I, using pancreatic islets from the healthy donors that were treated with the cytokines, I was able to show that most likely the same mechanism also present in the human. But the main thing is to show functionally that BHB really promotes mitophagy. And for this purpose, I use live imaging. So here the human islets, they were loaded with mitotrochic green that shows you all mitochondria regardless of mitochondrial potential. What is important for studying mitophagy, because actually mitochondria that lose mitochondrial potential, they undergo mitophagy. So in blue, I put laser tracker deprad. In red, I put the colocalization of these two dyes, actually showing you the mitochondria ending up in the lysosomes, actually the last step of mitophagy. And so I was able to show that really BHB promotes mitophagy. But so like adding additional fluorescent dyes to the same protocol, you can get much more information. So here I added mitosox, and which shows the ROS production in mitochondria. But using it together with mitotracker green and lysotracker deprad, you can see not only the bulk production of ROS in all mitochondria, but you can distinguish between like different mitochondria, for example. So using mitotracker green and lysotracker deprad, you can and mitosox, you can see what's going on with the ROS production in mitochondria that underwent mitophagy. And we have, so we can see that BHB promotes the clearing of mitochondria with high ROS production. You can better see from these experiments, so you can appreciate that when we treat cells with the BHB, there is much more white lysosomes. It's actually lysosomes when all our three dyes, red, green, and blue, colocalize together. We have these white lysosomes, and we see much more high-producing ROS mitochondria with high production of ROS ending up in the lysosomes. And when I block optineurin with a small molecule inhibitor, so we can prevent the accumulation of high ROS producing mitochondria in the lysosomes. Okay, so yeah, thanks for attention.
Stanis, that was great. Thank you for sharing that. And it's really, really nice to see the resolution of mitochondrial stains you're able to achieve, because I know that's by no means trivial to. I mean, I certainly wish that we were able to image mitochondria with that resolution over time. So we have, for the remaining part of the panel, I was going to open it up for questions. I will lead with one, and I have one for Jack. Not being an MRI scientist and knowing how impactful it can be to image real people's pancreases over time, I gather that by trying to normalize and standardize how you image across different sites, you're hoping to achieve enough reliability in your data that you're actually able to track changes in pancreas volume over time. Where are we at? Are we at a point where we can actually start to accurately follow longitudinally the progression of loss of insulin over time and start to pick up subtle changes in pancreas volume before or early on in the process? Because you showed an example, obviously, of somebody who had lived without beta cells for some time. But if you were to use this and roll this out as a longitudinal tool to try and monitor people before they present with diabetes or prediabetes, how far off are we from being able to do that?
So I think we're getting really close there. So we've done a lot of work kind of quantifying the test, retest, repeatability of these MR measurements, because we were worried, maybe the pancreas size in a person just grows and shrinks over time. And there's some data from UK Biobank that there may be some diurnal changes where the pancreas over the course of the day grows and shrinks a little bit. We don't see big changes. We typically see about a 5% variation. If you stick somebody in the same scanner, scan them, bring them back the next day, scan their pancreas size. We were really more concerned about reproducibility across different sites because we started working with Type 1 Diabetes TrialNet, started working across several different diabetes centers and wanted to make sure that we could kind of keep our data consistent across these different sites. And so we were able to get similar consistency across those sites and then followed people over time. So these kind of longitudinal scans you're talking about. And what we found is that some people, 4 or 5 years before they were diagnosed with T1D, their pancreas size started decreasing, maybe at about 10% per year. It tends to happen earlier than we see changes in their glucose tolerance. So it's not true for everyone, though. Some people, their pancreas size would be kind of normal until just prior to diagnosis. So I think it kind of speaks to probably a lot of heterogeneity in these populations. I think that endotype has kind of become the kind of preferred nomenclature there. And I think that we really need to start to think about these differences among people and how imaging will reflect those differences as well.
Great, thank you. And a quick follow-up. I mean, when you talk about reproducibility across sites, are you sending the same people to different sites and have the same pancreas image?
That's right. So we had 5 people who came to Austin. They went to Nashville. They went to Denver. They got caught in a snowstorm up in Chicago in the wintertime there. But same exact pancreas. And I was actually surprised not only those measurements of their pancreas volume, which are relatively easy, but also some of the more advanced measurements. Diffusion weighted MRI tells us about how water molecules move around, and those were also relatively consistent across site. Yeah, I was pleasantly surprised by how reproducible they were.
That's great and essential in order to be able to actually roll this out and be able to depend on this and make clinical decisions, or at least be able to add accuracy to measuring the progression of disease. And I want to take a moment to also give a shout out to Nicholas Mamrak, who's joined us from JDRF, who already chimed in with his thoughts and is acutely aware of the fact that they're listening here not just to learn from us, but also learn from us where our challenges are and what they might be able to do to facilitate the direction that the field is going. Nicholas, don't want to put you on the spot, but if you want to take a moment to kind of reflect on what you've heard and what you've learned from not just the people on this call, but in general from your conversations with people who try and leverage imaging to understand type 1 diabetes, and provide us with your thoughts on where you see us going next and where you hope that the field is taking this next.
Yeah, absolutely. Thank you. I will echo a comment that I believe Julia made earlier about cell type specificity and trying to understand targeting individual cell types. I think this is sort of one of the most pressing questions, right? And both from an imaging perspective, if we can get specific imaging of beta cells versus alpha cells, for example, this will of course tell us a lot about the natural progression, all of the interesting molecular and cell-cell communications that we've seen in presentations here today, but also thinking about how we deliver therapies, right? So if we have, for example, regenerative therapy, a pro-proliferative therapy, we need to make sure that that targeting is really sound. And so I think this is where that specificity comes in. I think in addition to that, some of the more translationally focused approaches like Jack's working on, that signal to noise ratio, which is a little bit limiting in some aspects, could benefit greatly from additional specificity, right? But kind of boosting that signal, or at least de-targeting and reducing the background noise. And I think the way we get there is fueled largely by discovery efforts, right? So single cell RNA sequencing, spatial transcriptomics, really digging into some of the molecular phenotypes, understanding what makes either beta cells, and improving sort of the cell specific markers that we have, I think kind of ties a lot of these questions in together. And, NPOD was mentioned already once or twice. And so I think, taking advantage of some of these resources that JDRF, now Breakthrough T1D, the name change, is, supported is really encouraging.
Great. Thank you, Nicholas, for sharing that. And yes, I'm aware of the name change. And yes, it's going to take a little bit of time for, I think, all of us to kind of change our habits and refer to JDRF as Breakthrough Type 1. And thanks for sharing your perspectives. I think it's really helpful to kind of have these conversations, big picture conversations, to kind of see where we are, what the challenges are, and maybe how we can work together in trying to advance us forward as a field, rather than to try and tackle these all at once. So we actually have a few people who submitted questions to the panel ahead of time, and I want to tackle at least one of them, which is one that we think about a lot in the lab and haven't quite figured out how to do it. And that's from Phil Packney from Novo Nordisk. He's asking, curious to hear what the panelists think about the advent of spatial omics and what it can do to resolve currently unanswered questions, presumably broadly relevant to the islets and type 1 diabetes. Who wants to volunteer and tackle that one?
Yeah, I mean, I said it already at the end of my presentation that it would help us a lot, definitely. But I also think that at the moment, we're not right there. So it's a really great technology, but I don't think we're there yet that we can tackle individual cells and not 3D. So for example, for the platform that many of us are using with the slices, you have different cell layers, but getting the cells from the individual cell layers after imaging, combining functional studies with this quantitative, as you said in the beginning, that would be amazing. And it would help us so much to understand, because we can only tackle so many things with functional imaging, right? If we combine this with the transcriptomics or proteomics or whatever, that would give us a such bigger picture and help us understand what's going on.
Can I just add in, is this currently happening in the neural field yet? Have they got into this whole spatial omics following the imaging? Or are they just kind of tiptoeing into it as well?
I actually saw a presentation last year by a neuroscientist on our campus at UCSD who showed how they combine calcium imaging in individual cells and spatial transcriptomics. So I'm hunting him now to get a collaboration.
Great.
Yeah, I mean, it's a challenge, right? So we can do we can do imaging with single cell resolution. We can do sequencing with single cell resolution at multiple omic scales. But getting those two things to work on the same cells in the same way that we're currently do post hoc imaging, like both Vira and Julia showed very nicely, that's, I think, a major challenge. I think it's doable, but I think I think getting the single cell resolution in the tissues is currently the bottleneck. I think it'll get resolved. And I think that's going to allow us to kind of close the loop between heterogeneity in gene expression that's been observed or concluded from single cell data with heterogeneity in behaviors that we see, and if there is a stable functional genomic correlate to that behavior or not. Yeah, it's something that I think many of us are trying to tackle, and I think we'll get there. But it's, in my opinion, it's one of the next challenges in the field. So the next 5 years will be interesting. Yeah, Alberto is kind of articulating what many of us know is one of the major challenges and one of the most impactful things we can do. If you could estimate beta cell mass and islet inflammation in living patients by imaging, would it ever be possible? And do we know how far physics and physical engineering can push us? Maybe this is a question for Jack again.
Yeah, I think it's a great question. So, prior to becoming more enmeshed in the MR world, I did a little work in PET, trying to get some of the VMAT2 targeted ligands for imaging beta cell mass. And it's a difficult problem, right? So, PET, if you can image a centimeter-sized tumor that's, expressing receptors that are 2,000-fold more enriched than the rest of the tissue, that's kind of what PET does a good job at. Imaging islets is a lot harder, right? They're smaller. The specificity of those targets, specifically for kind of the VMAT2 receptors, I think, fell well short of what was needed to differentiate exocrine from endocrine tissue. That doesn't, mean that there aren't other ligands or other targets that are more promising. But I think we just need to be more realistic about it and realize that physically it's just a more difficult problem than imaging, say, a tumor, or imaging, the striatum, something in neurology. So I don't think that it's physically impossible. I think we just need to appreciate how difficult a problem it is.
Sure, understood. And that may actually be like barriers in physics that will probably never meet the same resolution in those whole body pancreas images that we can achieve under the microscope. I mean, I think by extension, the same kind of trade-off exists on when you image live in vivo and when you image live under the microscope. I mean, different questions, I think, dictate the response best suited for that. It would be, Alberto, I fully agree, phenomenal if we could actually start to look at the single islet level and track mass accurately over time. Okay. There is a question in the chat that was posted ahead of time. And by the way, if anybody has questions, feel free to post them in the chat or raise your hand. I'm happy to give you the microphone in the final minutes of the webinar. But I'll start with this question. It's from Jesus Galaz Montoya, asking for any high resolution imaging efforts that look at phenotypes in patient samples at nanometer scale resolution with cryo-EM, etc. Electron microscopy tomography. And there's been some efforts in this. There's a study several years ago now from a Dutch group in Groningen who's done intact islets, whole islets, and just did tomography. And those data, I think, are probably available. And it was a recent paper that Monica just put in the chat that came out from Jeff Lichtman and Vira and Jane, who collaborated and really started to put together like mind-boggling amounts of data in just looking at high resolution in a small section, or in this case, the human cerebral cortex, reconstructed at nanoscale. If that can happen on a human brain, it can clearly happen on a pancreas. But it brings with it all kinds of questions, including questions around what do you do with all those data, and how do you like, there is so much information available there. Where do you start? So if you had access to data like that, what would the question? What would you be looking at first, right? I mean, I know that Vira will be looking at like alpha cells and beta cells that are next to each other and trying to see if there's ultrastructural differences between beta cells with and without alpha cell neighbors and maybe delta cell neighbors. But don't want to put words in your mouth, but I kind of imagined you wanted to go there. But what would we do if we had data that we know we can attain right now because the technology is right there? How would we leverage that?
Doesn't it seem, Mark, this is Monica, doesn't it seem that if you have a repository of this and different scientists from different disciplines can approach it, you know, they may draw different, you know, hypotheses and findings out of that said data. So the whole idea of a community-based data repository is so great. And if Pancreatless can pull that off and really engage with the community and build these kind of pipelines, it could be really powerful. I think, you know, the collaboration of that paper you referenced, the science paper with Jeff Lippman and Vera and Jane, the paper of petavoxel fragment of human cerebral cortex reconstruction at nanoscale resolution. They had like some big, you know, teams on there. Google, they had, you know, Harvard was on there, but they had Google, and they also had the David Allen Brain, you know, Institute. So bringing some of those big computational teams into some of these pancreatic imaging data might be something worth thinking about. And, you know, JDRF probably has some of those relationships. So just sort of throwing that out there.
No, I think you're absolutely right. And the challenge here is not getting the data. The data are now gettable, right? The challenge is like I don't know anybody in this call who's equipped to deal with that volume of data. And I don't think it's helpful or even possible for individual labs to create the data analysis infrastructure, the pipelines, but also the computational infrastructure to be able to handle those data. So I think in order for people to access those data in a meaningful way, you need to find a novel way of depositing the data publicly or behind whatever, some kind of link shared by anybody with a legitimate question to ask, so that they can then, I don't know, book time. Similar to how astrophysicists are booking time on like the fanciest microscopes, right? And say, hey, I got 2 hours next month. What are we gonna answer? And basically keep going back to these data sets and add samples as we go. Again, data sample availability and image acquisition, not that it's trivial, that's not the challenge here. The challenge here, how do you analyze this? How do you handle that sheer amount of data? And one final thing here that I think is interesting, and I know it's been done at some scale, is like community-based approaches to actually like pulling out data. There's been at a smaller scale efforts where data became publicly available and people could just, in the general public, sign up and say, hey, I'll just spend a couple hours on this. And actually just outsourcing and group sourcing data analysis is one interesting way to try and go, in addition to the obvious like AI, machine learning, algorithm development, to really to dig out of those data set stuff that might not be obvious or even quantifiable, but just sheer human hypothesis-driven effort. Anybody else chime in?
That is a phenomenal idea. And, you know, it just reminds me of AlphaFold.
Yeah. Yeah. We're at time. I'm happy to continue the discussion. I think this has been helpful. I certainly appreciate everybody who shared their current approaches, and I've learned a lot, and hopefully that will spur some follow-up conversations. Anybody else, any closing thoughts?
Just want to thank to Monica and Mark to putting this together. This has been a lot of fun.
Thank you all for taking the time out of your busy day, and you know, hats off for all you're achieving in the field of bioimaging of the pancreas and the pathology that comes along with that road to T1D. Thank you all again for taking the time, and I hope you have a great rest of your day and night in Paris. Bye for now.
Great. Thank you, everyone.
Thank you.